right. Well, welcome everyone to today's session titled Advances in Media for Cultivated Meat Production. My name is Elliot Swartz, and I'm a lead scientist at GFI specializing in cultivated meat. Cell culture media is one of the key technological pieces in cultivated meat production. Media has really enormous implications for the ultimate costs and environmental impacts of production, as well as understanding its formulation will be critical to the regulatory approval process. So I'm really pleased today to have such a diverse set of speakers and panelists show us some of the advances that have been made thus far, as well as discuss some of the challenges that still remain. So for today's agenda, we'll first hear from 3 approximately 5-minute flash presentations. We'll then move into a session with 2 longer talks, and then wrap up with a panel discussion. So I'd like to remind everyone before we begin of our participation guidelines. Please do share your thoughts in the chat throughout, but be respectful and courteous of others. If you are experiencing any technical difficulties, please navigate to the Expo tab on the left side of your screen and visit our help desk. So I want to save as much time as possible for the talks, so let's jump right into it. Our first speaker is Dr. Stephen Rees, the founder and CEO of Defined Bioscience. Stephen, the floor is yours.
Okay, thank you, Elliot. It looks like my audio is coming through. I'll assume yes.
Yes.
Excellent. So good morning, afternoon, and evening, GFC attendees, and thank you for attending this Cultured Meat seminar series. As Elliot said, my name is Stephen Rees, and I'm the CEO of Defined Bioscience. We're a new startup that just formed in the past 2 years, and we're based in San Diego, California. So I'll introduce Defined and some of its products and discuss some recent work that we've done with our collaborators in the cultured meat space. Next slide, please. So Defined Bioscience is a startup focused on streamlining the stem cell culture process. We aim to make stem cell-derived tissues easier to grow and produce for all interested scientists, and to make use of animal-free, fully defined formulations, trying to avoid proprietary, hidden, or unknown components. This includes a range of growth factors engineered for function and stability, media kits that are robust, scalable, and simple to use, and cell lines tailored for growth in fully defined conditions. Next slide. Our first products to hit the market this year include High-Def B8, which is an iPSC maintenance medium developed by the Burridge lab at Northwestern University, and FGF2G3, a thermostable variant of FGF2 that enables weekend-free culture with less material, developed by a lab at Masaryk University in the Czech Republic. Next slide, please. So B8 was developed by the Burridge lab at Northwestern, and you'll have a chance to hear from Paul during the session today. B8 was designed specifically to keep costs down while not sacrificing on the quality of cell maintenance. And while designed originally with the life science space in mind, we've found success in the cultivated meat space with B8 as well. B8 uses E8 essentially as a baseline, but replaces several of those components and optimizes the concentrations of others, leading to a medium with improved weekend-free capabilities and high passage counts. Next slide, please. FGF2G3 is a component that's included in B8 and is a variant Wildtype FGF2. It has 9 point mutations that bring its half 37 from less than a day to over 3 weeks. This allows for less material per liter of medium and improved stability to allow for fewer medium changes. Next slide. So as we said, B8 was originally designed with the life science space in mind, but the low cost implications are pretty clear for the cultured meat space. Andrew Stout, who's a graduate student at Tufts University in Medford, Massachusetts, and a member of David Kaplan's lab, tested B8's performance in cultured bovine satellite cells this past year, and found that this formulation worked quite well in cell culture. Specifically, Andrew conducted several passage tests of variants of B8, which included extra factors designed for bovine culture in mind, and he found that B8, when supplemented with albumin, allowed for a highly effective bovine satellites. With an additional component, B8 became Beefy9, on which the Kaplan lab just published this year. We're working with that lab now on further exploring this and developing some more products for downstream use. Next slide. We've also recently taken steps in the cultivated seafood space as well. So this year received a grant from GFI in collaboration with Reza Ovizapor and Lexi Dusher from Virginia Tech. You'll hear from Reza later today in the GFI grant session. This work with Reza focuses on differentiation of zebrafish and European lines for using the cultured meat space, specifically towards reagents and protocols that make culturing these cells easier, more robust, lower cost. Next slide, please. So it was a pleasure to speak with you all today. We're always interested in discussing more with those looking to make changes to their stem cell culture workflows. Feel free to reach out during this conference or through our website, definedbioscience.com. Thanks for your time today, and enjoy the rest of the sessions. Thank you.
Thank you, Stephen, for that. It's really great to see a product available that academics and other researchers can really just jump into using right off the shelf. So next up is Rajesh Krishnamurthy, the Chief Business Officer of Laurus Bio. Rajesh, over to you.
Thank you, Elliot. Good morning, good afternoon, good evening, wherever you are. Thank you, GFI, for giving this opportunity. This topic is pretty close to my heart, especially on the media, reducing the media cost for the cultured meat and cultivated meat industry. Next slide. So before we go into, let me give you a context. We are Laurus Bio. We are one of the largest manufacturers of animal origin-free media components for the media industry. We work with pretty much most of the media companies in the world, also directly with customers who use this. Next slide, please. So the way we do, we have world-class manufacturing capabilities, and we are scaling up our capacities. We recently inaugurated a 200,000 liter capacity here in Bangalore in India. And then we are, as we speak, planning for a 2 million liter production facility, which I will talk to you about how do we bring the cost down by scale. Next slide, please. So as we know, if you look at the operation cost for the cultivated meat industry, we see anywhere between 60 to 70 or even 80% of the operation cost coming from the media itself. And if you look at the media, almost 70 to 80% of the cost of media is driven by the proteins like albumins, the transferrins of the world, and also growth factors. So we play in approximately 50 to 60% of the total operation cost for the cultivated meat industry. So at Laurus Bio, we believe we have a lot to do to help cultivated meat industry to bring these products in the market efficiently. Next slide, please. So how do we do this? As I said, economies of scale. We are building large scale facilities as we speak. A 2 million liter with about 350,000 liter fermenters will give us a scale to bring the cost down. But we also know that just the scale is not enough. So someday we'll hit the wall. So we are working parallelly on the product innovation side, whether it's increasing the yield, changing our variants, and potency, the efficacy of some of these products. And most of the product innovation is happening on the process side. So downstream, especially if you look at our cost to make some of these products, most of the costs are driven through in the downstream purification process. Scale we are building, it will help us on the upstream. But on the downstream, we are doing a lot of thinking and thinking out of the box and eliminating or even changing some of the downstream process that we do for the pharma industry and see how we can look at some of those processes which are malleable to change it or eliminate it and reduce the significant cost for the cultivated meat industry. So we see that on the left side, you will see some of the contributions to reduce our cost. This is just an example of protein, not the growth factors. On the left side, the blue bars. Typically, what we see some of these products in today, what we sell in the pharma industry, the pricing anywhere between $50 to $200, $300 a gram. And our goal ultimately is to bring these costs down to almost $2 to $3 or even a target price of $1 per gram. So that's our ultimate goal. And what we have seen is, of course, the economies of scale gives you almost 40 to 50% at this time. As we build bigger capacities, we'll bring the cost down. But as I said, a lot of process innovation is happening as we speak. Yield improvements will give us some sort of a bump up here. So this is how we see from the protein side. But if you look at the growth factors today, it can go, anywhere between $500 to $10,000 a gram for the pharma industry, and that's completely not sustainable for the cultivated meat. So our goal as of now, our target is, can we bring that to kind of hundreds of dollars per gram? So that's what we're planning to do for the growth factors. So here's a graph. What we have done is we have developed several variants for the food application or the cultivated meat versions of our products. What you have on the left side is the standard pharma grade that we sell today. And if you look at that as 100, you can see that some of the variants and some of the process changes that we have done for the cultivated meat industry is not too far. We see about 15 to 20% reduction in the efficiency of some of these products. So, well, one of the variant even did better, so we kind of an outlier there. But we do believe about 15 to 20% reduction in the efficiency for the cultivated meat version, but the cost will be significantly lower. So we still have to look at the total cost of using this product versus the pharma grade. All right, next. So some of the graphs that we have developed internally, just to see on the blue, it's basically the pharma grade. One of the things that is happening is while we are working on the cultivated meat variants, it's also helping the pharma grade versions to bring the cost down for our pharma friends who are basically some of the vaccine, insulin, cancer drug, a lot of biologics companies that use our product. And on the bottom, the curve is where we want to target. Currently, we think some of these proteins, if you look at albumin, if we hit around 60 tons per year, 60 to 100 tons per year, is where we can go up to that single digit of $2 to $3 per gram. So this is the model that we are working on. There's a lot to do. Of course, it's not a straight line. Some of the variants we are working with, we need to rework. So it's a work in progress, but we are very confident and very hopeful to help and bring these costs down. So thank you. If you have any questions, please, you can put it on the chat room, or you can reach us by www.laurus.bio. Thank you.
Rajesh, thank you for that talk. It's really great to see some of the dramatic cost reductions that are already occurring and that will be planned in the future. The last flash talk for this session is from Marie Gibbons, a research scientist at VOW. Marie, over to you.
Hi, thank you so much. So I'm a research scientist at VOW, and I wanted to speak with you guys today about advancing cell culture media using definitive screening designs and high throughput robotics. Next slide, please. So here at VOW, we are constantly scouring the earth to find hidden flavors, textures, and nutrients. And this involves testing a lot of different media combinations for a lot of different species and cell types. So in order to do that, we've been using a whole bunch of different types of designs of experiments. And I wanted to speak with you guys about one in particular called the definitive screening design. Next slide, please. So our definitive screening design, or also known as a DSD, what it does is it analyzes large numbers of factors to identify which ones are significant within a large pool. It requires at least 4 factors, so you have to be able to have 4 factors that you're interested in, and then it tests 3 different levels. So let's say we wanted to look at 4 different factors within the BF9 media. In order to do that, we would need to test out 3 different levels. So let's say 10 nanograms, 50 nanograms, and 100 nanograms. the number of conditions that would end up being in the DSD is just 2x plus 1. So if we wanted to look at 12 factors, it would be 2 times 12, which is 24, plus 1, which is 25. And then in order to identify significance, you're able to get information around the main effects of each factor. So looking at the individual impact each factor has. Also, it looks at secondary interactions. So when 2 factors interact with each other, perhaps something like FGF and TGF-beta have a more significant impact when they're combined versus when they are when they're added in individually. And then they also look at quadratic effects, or what I like to call Goldilocks data. For example, let's say we're playing with FGF at 10, 50, and 100 nanograms. Maybe 10 nanograms is just not enough, and then 100 nanograms is too much. If we're able to also look at that 50 nanograms, and that's really a sweet spot there, the DSD will actually be able to tell us that data. So for those of you who are already pretty familiar with design of experiments, some of the benefits over fractional factorials and placket-Bermans that DSD has is that there are no co-founding between main effects and secondary interactions. So you can be sure that the secondary interactions that you're looking at are not due to just one of the components within that secondary interaction masking the other one. And then also these quadratic effects, or the Goldilocks data, is something that you can see with the DSDs, but not necessarily with your fractional factorials or your placket-Berman designs. So let's say that we wanted to look at the main effects, the secondary interactions, and the quadratic effects of only 6 factors. If we go to the next slide, we'll see that this is how many we would need. We would end up needing around 729 conditions just to look at 6 different factors for one species, such as a kangaroo, and one cell type. And this is if we decided that we didn't want to do any sort of design of experiments. Now, on the other hand, if we decided that we wanted to take that same number of conditions and use a DSD, we can go to the next slide. You'll see that we can more than double the amount of factors that we're interested in to 13. We can increase the number of species that we want to study up to 9. And then for each one of those species, instead of just looking at one different cell type, we can look at 3 different cell types. So now we can go ahead to the next slide. So I know what you guys are thinking. 729 conditions is still a lot of conditions. In fact, if you estimate that that each condition is around 2.5 hours of work, and then we multiply that by 729, we're looking at around 800 hours of work. And if each scientist is putting about 40 hours of work into their week, we would need around 45.5 scientists in order to look at all of these conditions. However, if you do this by throwing some automation into the mix, we can go into the next slide. We've done some calculations with our automation platform, which we lovingly refer to as Ranger, and each condition has now been dropped down to around 4 minutes of a scientist's time, and 4 times 729 ends up being around 48 hours, or a little more than 1 scientist needed to conduct all of these experiments. So if we go to the next slide. Oh, next slide. Oh, thank you. In conclusion, by using definitive screening designs and high throughput robotics, we can achieve around a 50x increase in capacity and a 40x decrease in labor. Next slide, please. Thank you guys all so much. And if you're interested in learning more about what we do, please check out our website.
Thanks so much, Marie, for that exciting presentation and progress at VOW. I think really accelerating experimental workflows with automation will be extremely important to rapidly iterate in this field. So we'll now move into a pair of longer talks. We should have time now at the end of these talks, hopefully, to field a few questions. So do drop those in the chat throughout, which I will be monitoring. And I will now introduce Dr. Paul Burridge, assistant professor at Northwestern University. Paul, the stage is yours.
Thank you. So first of all, I apologize for being the academic here and taking a longer talk, but hopefully I will try and make this reasonably concise. So next slide. That was some cardiomyocytes cycling calcium for you. So I have a stem cell lab here at Northwestern, and we're a very large scale. So probably 6 years ago, we really wanted to answer this question of, for a lab as large as ours, what can we do about media cost? And of course, many of you know, one of the most expensive components in these media that we use, right from the 1950s, was this component FBS, fetal bovine serum. So over time in the human embryonic stem cell and then human iPS cell field, we started with a very standard media, which is DMEM with 10% FBS, moving to a commercial product from Gibco called knockout serum replacement. And then over time, this has evolved to albumin alternatives, such as a recombinant albumin. And currently in the field, we're at the level where we have a chemically defined media. Now, this term is really commonly misused. Chemically defined means you're not allowed to have any albumin in there or any products that are non-recombinant because they may have other things that come along with them. Obviously, for us, the ultimate ambition is to move to protein-free because that will eliminate, as you've already heard, some of the costs to do with recombinant growth factors and other recombinant proteins that we use within these media. Now, the rationale for this was that the simpler the media, the more we can understand the mechanisms of signaling in these cells. But actually, practically, and not very popular in the academic literature, is that we wanted to improve the robustness and reduce the cost. Now, that's really important because academic scientists are terrible at saying my protocol is not very robust. And they also very rarely talk about reducing cost. Indeed, I think our paper was probably the first to actually kind of tackle saying, like, this is too expensive, and therefore that's a limitation. Okay, next slide. So on the left is some images from Jamie Thompson's first paper from 1998, so those are the first human embryonic stem cells, and you can see, so they're cultured in DMEM with FBS on a mouse feeder layer. And then on the right are some iPS cells cultured in a defined media with a recombinant matrix. I think on the right, you can see there some of the complexities of iPS cells. Can you see they have the pseudopodia? They actually reach out to each other to form colonies. You'd think it would be the other way around and the individual cells would move into a colony, but actually it's the reverse. So next slide. So this is a very busy slide, but this is the history of iPS cell culture media. So on the left, we have Jamie Thompson's FBS containing media moving into the KSR media. Initially, they were KSR media conditioned on these mouse fibroblasts, so getting all of the growth factors from them. And then slowly over time, figuring out things such as high levels of FGF2 were advantageous, that TGF-beta was important, or activin A. And then slowly progressing from using KSR. And then in the middle, we have using BSA, bovine serum albumin or human serum albumin. And then on the right, you can start to see the true chemically defined media. So what I'm trying to show you here is that for us to figure out the signaling and the way to control human iPS cell growth in a very defined manner, it took 42 papers from about 35 labs. So if you think the average academic paper costs about $300,000, you're looking at about 12 million to be able to move us through that entire progression. And many different methodologies have been used to discover these kind of intrinsic facts within this. So in the bottom right there, you can see one media called Ideal, which was developed using a design of experiments methodology, which actually in this particular model wasn't very successful, in part because the design of experiments method requires multiple repeats, which can be pretty laborious for us. So next slide. So the signaling in these cells is actually, we now know, quite simple. We're not allowed to affect BMP4 either up or down. The cells require that to be in a mid phase. Activin TGF-beta and nodal, each can work. The main signal is FGF2, and indeed, the reason why there wasn't more progress early on in the field is that the human iPS cells or human embryonic stem cells require a really high level of FGF2. And when we first started culturing these cells, indeed, the thing that really held us back was FGF2 was very, very expensive. So indeed, when I'm going to talk about the E8 publication, so the first chemically defined media, and I actually had, the day that was published was the day I made that media. We had all of the components available. It's just none of us were willing to use FGF2 at 100 nanograms because it was incredibly expensive. So that was, for me, my first lesson that the cost of growth factors actually is preventing the science progressing. Therefore, developing my desire to fix that issue. The other factors are involved are IGF or insulin. And then finally, Wnt signaling is also somewhat enigmatic, that you can use a positive Wnt signal to maintain pluripotency, but actually it ruins the differentiation, the same with inhibiting that pathway. So what I'm trying to tell you is for every different cell type that we're going to be culturing, there are a set of rules that we can follow, but actually within that set of rules, there are some complexities that potentially can take a lot of time to figure out. So next slide. So this is what happened in the field. I mentioned we started off with this KSR-based media, which was very common. It's commonly called human ES cell media that contained KSR as the most expensive component, a very low level of FGF2. Then there was this rapid development of commercial media, moving into one called MTESA, and then Essential 8, and then most recently, commonly in the human iPS cell field, media called StemFlex and Essential 8 Flex. I would actually say most labs are still using MTESA, even though it's been a long time since that was developed. And as you can see, these are prohibitively expensive. And for an academic scientist, very frustrating, because I'm going to show you that these media were all developed from an academic lab, so we actually know how to use them, know how to make them, but yet somehow Stem Cell Technologies is still willing to charge you $500 a liter for it. So next slide. So MTESA, so that was the first transition from using a KSR-based media into what was a defined media. This is probably not the best way of showing you it, but the question was all about the method for developing it, and it was a very strange method. So they essentially figured out what receptors the cells have and then added in the ligands for those receptors. And they found lots of interesting things that seemed to work, but actually all that did is add up a huge amount of complexity to the media. This idea of, how do you take something as complex as a KSR media on MEF and then transition it into a defined media? Actually, all of the routes that have been taken using a kind of intellectual approach have not been very successful, and the ones that have been really successful have been much more empirical, testing things and seeing what the result is. So next slide. So that's where we ended up with. So we moved from a KSR to MTESA, which was very complicated but defined, and then looking at the components in MTESA and trying to figure out what the issues were and what could be eliminated. And one of the things you'll see here at the top left is they seem to have a lot of issues with albumin from different batches. The performance seemed to vary quite a lot. So they considered albumin to be a variable. Obviously, it comes from lots of different sources, and this is not a recombinant albumin they're talking about. So this was their major issue with MTESA. The albumin was variable, giving them different results. And then, to cut a long story short and a big project into a single sentence, they found one of the components, which is very commonly used in cell culture, called 2-mercaptoethanol, an antioxidant. Once you took the BSA away, or the albumin away, the mercaptoethanol was toxic. So what you needed to do was eliminate the two things simultaneously. And then, so they figured this out. Great work. Seems kind of obvious in retrospect. And then they basically developed further versions of TESA or MTESA. by seeing which components were necessary, taking out the insulin or the FGF2. Basically, a lot of plus minus experiments to eventually, and as you can see, each different thing that they've tried taking out here, they actually did it with a different type of experiment. A survival index, a proliferation, a combination of both, a cell number, you know, in 2011, like, this was the best media. This was the most optimized and simple media. They called it essential 8 essential factors. And then this was the cutting edge technology. Like I say, when this was published, we started making it with a few of us in the field that were interested. I was working on a defined media at the time. You know, so to have it published was a little frustrating, but also the fact that it was pretty simple and it moved us forward a long way was really interesting to us. But I hope you can see that although they have got a media that contains only 8 components, the method of how they did the optimization is not very convincing. There's no dose optimization. There's no, you know, multiple subtractive experiments. Certainly not as sophisticated as you'd hope, especially for a product that ends up being commercialized. So next slide. So we started working on this. So, you know, I hope you're all aware that an academic paper that came out, you know, was available 2 years ago actually means that we really completed it 5 years ago. So there's been a lot of progress within my lab on this since, a lot more to come in the future. But essentially we started with the E8 formula and the concentrations they had in that paper. That's a retroactive simplification of what we actually did. We actually looked at all of the different chemically defined media. We tried them all. We tried different combinations of them. This is all in the paper. And we actually figured out that E8 pretty much had the optimal concentrations and was the best of those. We developed this assay where you seed the cells at very low density and then you grow them for a long time with the assumption that low density means low clonality and therefore that's a higher pressure on the cells. So basically to try and develop a media that's a little more robust rather than our standard much shorter experiment. And most importantly, we took this huge leap of faith that if a cell is alive, that it's pluripotent and, you know, is a stem cell. That's actually, those two things are not necessarily connected. We're obviously a differentiation lab, so that's important for us that they're still pluripotent so that they differentiate. But so we now had an assay where we could very easily change different concentrations of things and look at what effect that has on the cells out the other end, and their proliferation rate and their survival. So next slide. Okay, so this is where we ended up using this assay. You can see just about, if you can see to insulin, the ascorbic acid, transferrin, all the different components, and we optimized them. So if you just step back, you can see basically the gray bars are, the solid gray bar is the starting concentration and the striped one is our ending concentration. Some things went up, some things went down, but pretty much it stayed where it was. Intriguingly, I mean, you can kind of tell from these graphs, actually, it's not 8 essential components. There's actually only 4 additional components. So just 5 things, including the basal media. That's the risk you take if you call a media something like essential. It's quite easy to show that some things are not essential. So, you know, we did this long single passage assay and, you know, with commercial suppliers, we took the media cost from about $500 a liter down to about $60 a liter. And we used this media for a long time and it works just fine. So next slide. So then we worked out, actually, you need a more sophisticated assay. And this is where things started to become, you know, you've just heard from Marie that, you know, a machine can do this. I wish we had a machine to do this 5-passage assay. It's an incredible amount of work. I had a phenomenal technician who just really labored away. So this is 5 passages. Each of those are 4 days. So, you know, you end up with this. The actual entire process is about 25 days, including getting the cells set up and then assaying out the end. And each one of those dots is a completely separate experiment. So then we did this kind of higher level of optimization in a long-term assay starting with our kind of optimized version and ending up with what we now call B8, which in the lab, the joke is it's better 8 rather than essential. And I don't think better is very quantifiable, but certainly for us, works just fine. And you can see B8 has an additional growth factor, neuregulin, that comes from one of those other medias that we tested. Most other things stayed pretty much the same. But on the right, you can see that this was now our new ambition, was anything where we could reduce the cost, because, you know, once you reduce the level of something, to some extent, the cells can then adapt. And insulin's a good example. We showed previously that 20 is right. And actually, if you reduce it to 5, over those 5 passages, the cells learn to deal with that, and then 5 is fine. So that's one of our most expensive components in the media, and now we can use a quarter of it. And then when it comes to the growth factors, we decided to produce those in-house. So next slide, please. Not trivial. There's a reason why people don't make their own growth factors, but actually it turned out not to be too bad whatsoever. So we started off with this FGF2. In the E8 paper, they talked about making one with a single point mutation called K128N. As you heard from Stephen Rees, there's a paper published from a group in the Czech Republic that had looked at this more complex version. So we tried them all. We made 5 different versions of this recombinant growth factor, trying to look for stability. FGF2 is not stable in the media. The more stable it is, the less media changes we have to do. And this is essentially how we got to the weekend-free protocol. Week-free is a bit of a salesmanship side of things. Actually, all media can work weekend-free. It's just about how well they work. If you see the cells at a low density, weekend-free works with just about any media. It's about when the cells are at kind of standard high density, whether it's still successful. So next slide. So essentially, we published this in the paper, or back one. We've published this in the paper. We actually have a very simple system. You make a liter of E. coli, you lyse the cells and pull them out using a spin column, because this has a tag. So very, very easy to do. So next slide. And then this is the results of the weekend-free protocol. Or it's my teaching slides. We got a little bit of math involved there. For IPS cells, we split them 1 to 20, so that's a 22-hour population doubling time. And then here you can see, so this is the bars in the middle show you the cost of commercial media, about 500, and then B8 down at about $10 a liter. That $10 a liter number is incredibly conservative, because our reviewers were giving us a really hard time about including labor within that. Actually, within our own lab, the current version of it that we're using is logarithmically cheaper. We've made it very, very cost effective now, even within a lab scale. So like I say, that cost is conservative and includes the cost of a bottle of basal media, which is the majority of it. That little aliquot, just pretty much any lab following our protocol, which is on the website, can get that single aliquot down to below $3 pretty easily without even having to buy any equipment. If you start to actually in-house it all properly, it comes down much further. So next slide. So this is as many of my group as I can get to turn up for a photo. There's obviously a few more than that. All of the work I'm talking about, we've never had it funded. Very happy for people to be involved in funding these kinds of projects in the lab. It's always just has to be done of startup money. NIH doesn't really fund this kind of stuff. So yeah, sure. I'm happy to take any questions if I have any time.
Paul, thanks so much for telling that story. I think it's really important for people to understand. I think, as mentioned in the comment there, you know, the tour de force efforts that it takes to iterate upon these designs, but also the demonstration that, you know, truly these medium formulations do not need to cost as much as they do. So unfortunately, we don't have time for questions, but I will drop 1 or 2 in the chat if you do want to entertain them, and maybe you can answer to any others in the chat. But we'll move forward to keep on time. And so our final long talk will be from Dr. Suzanne Weigle, who's the investment manager of alternative proteins at Nutreco NuFrontiers. Suzanne, it's all yours.
Thanks very much, Elliot. You're welcome, everyone. It's an honor to present here today at the Good Food Conference, and I believe I will add a view to the session that comes from a very different point. I'm investment manager for alternative food proteins at Nutreco, that is an animal feed company and has a corporate venture arm. And we are partnering with our portfolio companies to actually build a business in cell culture media, but from the basal media side. So we are talking today about the nutrition growth factors. We know very important, very strong cost driver, but we believe the more this industry is upscaling, the more important it becomes that basal media is very cost effective. Next slide, please. So Nutreco is a global leader in animal nutrition and aquaculture feed. We are farming protein sustainably. That is a very, very important part of our culture. And while investing in companies in the alternative protein space and learning about the space, we have figured that actually the science of feeding animals that we are applying on a daily basis will be vital to feed cells sustainably. Next slide. A few words about Nutreco as a company. We are a global company headquartered in the Netherlands. We are family owned, which allows for a broader vision and some future investments. We have two business units. Trout Nutrition is our business unit that takes care of the land-based animal nutrition. So you can imagine we are feeding any animal that runs around, cow, pigs, chickens, but also pets and even horses. And we have strong aquaculture feed division, Scratching, that is headquartered in Norway. If you are not yet a vegan and you like salmon, every third salmon in the world is fed by Scratching. So very large global operations, more than 100 production plants, lots of employees, and specifically, and I think this justifies our role also in upscaling here, we produce 9 million tons of feed products each year. Next slide, please. We are in the same challenge. So we see, of course, the importance that animal protein production plays today, and we are welcoming very much the rise of a new industry in alternative proteins, because we really believe we need to change how we are doing things if we want to feed even more people in 2050. Next slide, please. So our mission on the animal feed side, but also justifying our engagement in alternative proteins, really comes from our core mission, and that is feeding the future. So sustainably producing protein in whatever way is very much at the heart of our mission. And this is why we are engaged in this space. Next slide, please. We have invested in several companies. So on the cell culture side, we are investor in Blue Nalu, San Diego-based seafood, and Mosa Meat, Netherlands-based, working on beef. We are also investing into fermentation-based companies. Our recent investment is Enough, formerly 3F Bio, that produces mycoprotein with a zero waste process. Maybe not known to everyone, alternative proteins are not only important for food, but they're actually also very important for feed. So we have a partnership with Calverdi, which is the feed company, which is the Lisa Dyson's air protein in the food space. And we are looking here at producing protein ecologically sustainable and efficient for aquaculture feed. Why are those companies partnering with us? This really very much lies in our capabilities. We are one of the largest users in the world, really, of many, many ingredients, among those vitamins, amino acids, trace minerals, but also agricultural raw materials. So we have grand ingredient expertise, large procurement power, supply chain very well set up, and of course, large-scale manufacturing capabilities. Nevertheless, feed is very highly regulated, so we also provide a high standard quality system combined with very well regulatory expertise, and which I find extremely important for this space here. We practice daily precision nutrition expertise. This means how to feed actually animals efficiently to get to a very good feed to food conversion rate. On top, to fulfill our mission, environmental sustainability metrics are very important part of our capabilities. Next slide, please. So what are we looking at here, actually? So when you think now basal media for growing cells and scaling up the cell cultured industry, we are really talking vast production of media in the end. Feed today is produced at 1 billion tons per year. And of course, the whole aim is to become more efficient with cultivated meat. But nevertheless, if you want to produce 10 million tons of cultivated meat and seafood, as predicted by a McKinsey report in 2030, you need vast amounts of inputs. And so what we see with the current cell culture media, well, it was developed for a totally different industry. Costs do not play a role. 2% of biological drug production costs are allocated with media, so that is really not very important. We have heard a lot, highly defined, single molecule ingredients, extremely complex mixtures at high costs, and media is optimized very much, but always for drug production output, not really for costs. In general, a low volume, high margin production business. Animal feed is exactly the opposite. Animal feed was developed to actually produce food, so which is what we all want to do. The costs for animal feed at a farm are quite substantial, 40 to 70% usually, and therefore there has been for decades already a strong focus on optimizing the nutrition at least cost, actually with renewable and cost competitive ingredients, because of course the massive amounts that are produced each year need to be renewed. Efficient conversion of feed into food, and in general it's a high volume, low margin product. We want to bridge that. So we want to actually define cell feed, cell culture media for producing food. And what we need from today's media is mainly the quality. Of course, it must be suitable for cell culture. It must work for cell culture. Your cells cannot die from microbial contamination. But on the other hand side, we need it at low cost, high volume, food safe, and really with an efficient feed to food conversion. Next slide, please. So we see that 3 principles that are practiced every day in our animal feed production are important to apply to basal media. First of all, well, media, pharma-grade ingredients. We heard it yesterday also in the discussion with Mark Post and Uma Valetti, is a no-go. We need to switch those ingredients to food grade and potentially have even potential to switch them to feed grade. In principle, most of those basal media ingredients, like here typical DMMF12 formulation, are already applied in animal feed and also in food production. It's kind of like the vitamin pill for the cow, and it's called feed premix. And so many of these are really available in both grades. And that's the first transition that we need to make. That will not be enough though. Especially on the amino acid parts, we need solutions to not put in single ingredients, but we need to look back and look at how animal feed in principle is done. The majority of nutrients really are provided through agricultural crops, through wheat or corn, soy protein, of course, other proteins. A vast variety of agricultural inputs is used for that. And then you put in the vitamin pill, some amino acids, minerals in addition in this premix, and you have a complete feed. This is the next step that we need to achieve for cell culture media to bring the costs down. And then thirdly, the principle of precision nutrition needs to be established and implemented at large scale for producing this basal media. Animal feed compositions are currently constantly optimized for maximal output. And what we are doing is we are linking really economic models to the procurement, to the supply chain with nutritional ingredient parameters, nutritional requirements for feeding the species, and with sustainability metrics. All these principles can be adapted to cell feed. And we are on a journey together with our partners to do so. Next slide, please. When we look at the first step, just switching normal media formulation, whatever it is for each cultivated meat company, to a food grade media formulation. Well, we have sourced food grade amino acids, and in our partnerships have shown that it works. So it's not really rocket science. Here's an example where a pharma grade media was tested on real cell system, and the amino acids were exchanged with food grade amino acids, and it works very nicely. So there are no, of course, one has to look at each ingredient and each cell line and so on, but there is in principle no reason to believe why the first step could not be done. Next slide, please. If you want to look long term, feed grade ingredients are quite interesting to consider. Of course, we have regulatory situations, and as we are all taking just the first regulatory hurdle, it's a natural thing to stick first to food grade media. But ultimately, we want to make sure that our supply chains have really the capability to flexibly use ingredients to bring down costs and to also accommodate for really large volumes. And this is what you find in the feed industry. In general, feed-grade ingredients very often show similar purity profiles to food-grade ingredients. And so, therefore, you can aim for further cost reduction, of course, in a dialogue with regulatory authorities, if you are able to include these in your media. Here's one example. Feed-grade glucose works as well in a media like the pharma-grade one. So we within Nutreco currently are performing a safety and risk assessment on the various ingredients to compare food versus feed ingredients. Where could this be applicable and advisable and where not? Next slide, please. Here just a short illustration how one can think about costs. An example here of an amino acid, L-lysine, just average market price at small scale per kilogram, really just from official websites. Large scale prices are, of course, much lower. And what you see is you have a large pharma grade price over 200 euros. 10 times cheaper is already food grade. So there are a lot of cost benefits to be expected. But at feed grade, you have another 10 times reduction. Prices do differ, of course, between ingredients and scale levels. Also, not every ingredient will be perfectly suited on feed side and food side, but we are performing an analysis right now to identify those ingredients that could benefit from flexibility later on when there's really a lot of pressure through large-scale production. Next slide. But as mentioned, that is only the first step for making basal media more cost effective. It's very important to specifically exchange those 20 or even more amino acids that are currently included in basal media as single molecular ingredients by hydrolysates of agricultural protein materials. So that is an important step both economically but also environmentally, because it's environmentally not very friendly to do those 20 ingredients separately. So as our aquaculture division has some experience with hydrolysate, we are looking into this topic and using our different protein sources that are very important in aquaculture feed and used very readily to analyze what could work for cell cultured protein production here. There are interesting synergies potentially with the aquaculture feed production because of the vast amounts that they need, different ingredients, but also because they are constantly looking into innovative protein ingredients. Best example is the KGaA relationship, where we are producing bacterial protein based on a gaseous fermentation, which is very sustainable. And so there are some synergies between these two businesses. Next slide, please. This all works only with a large supply chain. So it's very important to bundle actually these requirements for the various ingredients. Let it be macros, which are these agricultural raw materials, or let it be micros, the vitamins, minerals, amino acids or so, because otherwise you're not really getting at the costs that you need to upscale largely. Just as an example here, Nutreco procures 8 million metric tons of crop material and 1.6 million metric tons of micro ingredients. So that is really a strong global and, by the way, also local supply chain, which with many supplier relationships. Of course, the quality is also very important. There is a dedicated quality assurance team behind this to ensure that only the right ingredients are procured. And this is all done by a network of more than 100 procurement and supply chain professionals. Next slide, please. I personally still think most exciting is really the third principle, translating the precision nutrition that is used daily in making our animal feed products for cell feed, for the cell culture media here. And this is a system that really integrates information on each single ingredient, like even the raw material, quality control, how nutritious are the ingredients that are coming in and that are available at that very moment when this feed formulation is supposed to be made, with databases that include all the nutritional values, bacterial characteristics, and many more aspects to be able to come up with the best low-cost formulation that fulfills a specific nutritional need. The specific nutritional need is there is a lot of animal nutrition know-how behind there in animal models. Every single ingredient is essentially studied, how is it transformed in a certain species into the product that you want to have later. Let it be meat, let it be dairy or eggs. And this is the core of such a precision nutrition model. But the linking to the supply chain in real time in the end, with varying prices on ingredients and raw materials, is very much key, so that you can really get least cost formulations. You then put that into certain economic models and find the best options there. What I personally find also very exciting, and I think this is ultra important to really deliver on the promise that the cultivated meat industry has, is we are linking this also to environmental sustainability criteria. So you can choose not only the least cost formulation, but you can choose that formulation that fulfills the nutritional need, but at the same time has a cost environmental benefit ratio that you choose. Next slide, please. Just a quick note on quality. Of course, we are talking here cell biology, and we are talking food production. So quality is of utmost importance. Quality is also of utmost importance in feed production itself. So as an example, Nutreco has really a complete quality assurance system called NuTrace that looks at all different kind of angles and makes sure that all ingredients and suppliers are perfectly assessed, monitored and controlled. And also very importantly, it actually assesses the risks in the supply chain that at any time point there will be enough of a certain ingredient at the required quality without any problems. Because of course, each single ingredient should not only have one supplier and then suddenly you have a fire in the plant and you have no ingredients anymore, but you have really sophisticated system of many suppliers that are all quality assured and to deliver a standard that is with very low risk. This is also adaptable to the standards that the cell culture protein industry needs. And as we are speaking, this is currently done with the Nutreco. Next slide, please. That for me is also, always the most important, the sustainability and how can we really ensure that. Those inputs will also be very, very important for the environmental balance in the cell culture protein industry processes later on. And so what we have for animal feed is the system-wide feed print, where essentially we have a standard aligned environmental footprint database where each ingredient has a CO2 footprint. And that goes into our system for precision nutrition. As mentioned, then later on you get out footprint per kilogram feed, and then you have the option to choose which cost environmental benefit ratio do you want later on. It becomes very exciting when you link this to the actual cultured meat or seafood production process, when you can make sure that the overall footprint is also with that nutritional formulation optimized. Give you an example, if later on an addition of a certain amino acid maybe ends up with a much more productive growth in the bioreactor, then maybe although the feed itself is a little bit less good in its environmental aspects, the overall process would still be much better. So this link to the cell culture protein production process is very important too. Next slide.
Suzanne, I just want to ping you that we're a little at time, so if you can wrap up in the next 30 or 40 seconds, that would be great.
Yeah, and that's good because it is my last slide actually. So we believe here are a lot of principles really important for scaling the cell culture protein industry, starting with just food grade normal DMAM mixes, but at the right quality system and food standard production, going over using agriculture production up to precision nutrition. And if you are interested to join our journey, we would be very happy to get in contact. So any questions on the cell nutrition business or on also on investments, please don't hesitate to contact me. Thanks very much.
Excellent. Thank you so much for that presentation. I think it's really fascinating to rethink cell culture media as as we have done for decades in the animal feed industry. There were some questions in the chat, Suzanne, if you want to try to address those afterwards, but in the interest of time, we'll move forward into the panel session.
Thanks a lot.
So a reminder, people can also put questions in the chat throughout this panel session, and we'll try to monitor those and incorporate those throughout. I'll just sort of introduce some background to this session leading up to introducing our panelists. So it really was in 1882 when Signe Ringer created the first balanced salt solution that allowed for the culturing of tissues outside of the body for the first time. And it wasn't until around 50 years later when researchers began to piece together the fundamental requirements for cell growth that we've been hearing about today: the amino acids, glucose, vitamins, salts, and other growth factors that underlie cell culturing. So today's media formulations are these complex mixtures that typically contain dozens of individual components, where each of those are, as we heard, fine-tuned to the cell type being used in their downstream application. And as mentioned today, the cultivated meat is really addressing tackling this challenge head on by reimagining cell culture media formulations, as well as some of the origins of the media components. So recent survey data suggests that many cultivated meat companies have already achieved dramatic cost reductions, some as high as potentially 99% or more, but there are challenges that still remain in optimizing formulations and building out the supply chain for these inputs. So to help flesh out the challenges and the progress to date, I'm joined today by 3 accomplished panelists working to advance cell culture media for cultivated meat production. You all can come on screen now. The first is Dr. David Block, professor of chemical engineering at UC Davis and recipient of awards from the National Science Foundation, GFI, and New Harvest for work related to cultivated meat. Dr. Aletta Schnitzier, the R&D director of cultured meat at MilliporeSigma, which is the life science division of the global science and technology company Merck, KGaA, Darmstadt, Germany. And also Dr. Pallevi Srivastva, the head of cell culture media and process development at Wildtype, which is a US-based company developing cultivated salmon products. Welcome, everyone. So I think in order to sort of understand the advances that we are witnessing, I think it's important to first sort of provide a frame of reference for the audience for what cell culture media was when this industry began to materialize 7 years ago, and some of the remaining challenges. So Aletta, I'd like to start with you first. Over the last decade at MilliporeSigma, you've worked on many aspects of media optimization for stem cells, therapeutics, and now cultivated meat applications. So how did this prior experience help you understand the challenges in cell culture media that would be facing the cultivated meat industry? And what is the approach that MilliporeSigma is taking in media development for cultivated meat?
Sure. Yeah, thanks, Elliot. And first, just it's great to join you and Pallevi and David on this panel talk today. Probably the first thing that my experiences over the years at MilliporeSigma has taught me is that media development is a very iterative process. Formulations are constantly evolving, and not only with cell line developments, but also the changing manufacturing processes that are used. Years ago, I remember attending a media talk for a perfusion process that compared a few different formulations in conjunction with a TFF, a tangential flow filtration device. And the pharma company ended up selecting the final media not based on the productivity, which was quite similar between the offerings, but on the longer process time that one of the media formulation yielded before the filtration device was fouled. So, I mean, some formulations have high molecular weight components that can prevent aggregation, which is great in certain processes, but can really impact downstream processing. So, as a media developer, I'd say we really need to understand the whole process and how the media might impact all of the unit operations, in addition to the cellular responses and driving cell numbers. So the approach that we're taking to media development for this industry is pretty multifaceted. From the cell standpoint, we're working on myoblast cell line that's immortalized and suspension adapted, that hopefully represents the future state. And then we're also working collaboratively with partner cell banks from biopsy to understand the unique challenges and media requirements the industry will need. But I'd say the main thrust of our program currently is putting in place that toolbox of food grade material. So it was great to hear the previous talk, because it's so important to lower the cost of those raw materials, but in a way that both meets the regulatory and performance expectations. So, having to test the different sources and demonstrate performance, but also going through that whole qualification process so that we can implement these materials in cell culture media manufacturing at scale is really, the primary objective right now.
And I'm just curious, is this operation mostly in-house at MilliporeSigma? Do you work with some of the companies in the sector, academic labs? How are you going about, solving this sort of diverse set of challenges by leveraging some of the others that are working?
Yeah, all of the above. So my my new automation engineer just attended the UC Davis short course and is very excited to connect with some of the folks in your group, David. So there's already chatter going on about how we can improve our automation techniques. We're working with select culture meat producers to really understand their challenges. And then regarding our manufacturing, many of the components that we use, we do not manufacture in that part of the supply chain. Rather, we source them, test them, qualify them. And so obviously that's being done in collaboration with external folks. The manufacturing itself is in-house.
Great, thank you for those insights. David, I'd like to bring you into the conversation now. You have really extensive experience in fermentation process optimization, as well as these sort of experimental optimization methodologies, but perhaps a little bit newer to some of the challenges in animal cell culture. So what made cultivated meat an attractive challenge for you, and how's your lab working to optimize some of these formulations?
Yeah, first of all, thank you for inviting me to this panel. I've been working on fermentation optimization now for over 30 years, from first in industry, now in academia, and industry from a few milliliters up to 160,000 liter scale, making both pharmaceuticals and bulk chemicals using some of the raw materials that you heard about in the previous talk from Suzanne. So as a lot of the speakers have said already this morning, optimizing media is really time consuming and really expensive. And so when I got to Davis, we started working on methods, and there's some chat on this and some entries in the chat on this as we were going through this morning. We've focused on developing experimental optimization methods that are much more efficient than statistically designed experiments, which are already pretty good, but they kind of lose some of their efficacy when you get to large numbers of media ingredients or lots of interactions and things like that. So we've worked on methods that use artificial intelligence or machine learning to accumulate knowledge as you're going along, and some advanced optimization methods to suggest new experiments. And we showed probably more than 10 years ago on lactic acid bacteria fermentations that we could cut the number of experiments needed to get to an optimum in half. And more recently, we really were interested in applying this to cultivated meat because this is a huge hurdle for the industry to get to commercialization. And so we've improved some of our methods even further more recently with students like Zach Cosenza in my lab, and shown again 30 to 40% savings on experiments using these methods. I will say, while all those methods are really important, it's also very important to really understand your process and understand which nutrients are being used by the cells at any given point. So this kind of black box approach in parallel with a white box approach are probably the best way of moving forward.
And for some of the papers that you publish, are these methods just easily replicable by others in the sector? How do you sort of go about sharing those methods so others can adopt them?
Yeah, so we started publishing these methods over 10 years ago. Zach has published 2 papers recently. One is in press, the other one is out. One of our goals there is to make these methods robust enough so someone who's not an expert in artificial intelligence or advanced optimization methods can still use them. So many people in the audience may have used design of experiments software that's pretty easy to use. The goal for us by the end of Zach's Ph.D. is to get to the point where that can be done. if you have people, and it looks like there's some people on chat who are experts, and I know I've talked to someone in Aletta's group who is more an expert in this area, reading the papers and seeing the algorithms might be enough, but the hope is to get to the point where everybody can use these methods.
Yeah, that's super important and great to hear that that's the end goal there. Pallevi, I'd like to now bring you into the conversation. I think, developing media for seafood in particular presents unique challenges because most of the knowledge for media development, up until 10 years ago was human and rodent cells. So, recent prototypes that I've seen in the news and announcing your pilot scale facility, you've clearly made some progress on this particular point. So, can you describe how you've approached the challenge of media development for aquatic species like salmon, and discuss any of the advances that your team has made to date?
Sure. But before I begin, I thank you for such a great session. I enjoyed every talk today. It's great to be here. And regarding the question, I think I should really start from and note that when Wildtype started, there was no cell lines available for us to develop media for. So we really had to start from the very beginning and establish cell lines, and then at the same time understand how to culture them. A lot of historic work that was done was using a medium L15, which is a very lean medium. So as we were establishing these cell lines, we were also trying to understand how to grow them, what is the right media formulations. And we learned that the pH for them, for example, is very different from mammalian cultures. My background was mammalian culture, so it was very surprising for me to see the temperature range is very different from them. And we had to figure out like what plating densities, what surface works for them. So it was a lot of learning, but the challenge mostly was around, okay, you have to establish your lines, but at the same time work as they're evolving, and as the strain is evolving, you have to continuously change your formulation. So that was very tricky and challenging, I think. And so for that reason, I think a lot of development, or most of our R&D progress has been made just on that, doing parallel tasking of establishing the line, but at the same time establishing or developing a formulation for it.
So is the strategy that's taken in-house, is it mainly to work and focus on one line and optimizing there? Are there multiple lines that you run in parallel to really figure out, what works best? How does that process work for you?
We do have multiple cell lines, but there is one primary that we focus on for our product, but there are multiple strains and lines that have been developed that are in the pipeline, and we try out any formulation that has worked for our primary line. We try those out and screen them for the next set of lines, because once they're very similar, the aquatic species or salmon is very similar in the nutrient requirement. So we found that if we have the framework or the skeletal structure of the basal or media figured out, we can screen additional lines with it. Maybe not the best approach, but that's like the small team. I think that's what we are going with.
Excellent.
Elliot, if I can just add to that, what Pallevi is describing is always a problem when you're doing fermentation process development, because if the strain is evolving at the same time as the media, it makes a combinatorial optimization problem huge, right? And I think one of the ways you can do that is, and it sounds like this is something that Pallevi's group is doing, be very conservative about when you introduce new strains. Not every little iteration of a strain should be introduced into a process development program, and use very efficient methods. Come up with a media that's a starting point for each of these new strains, but you're still going to optimize or have to optimize around that. And there's not a one great solution to this, but it's always going to be a huge problem.
Yeah, thanks for that insight. I love bringing in the knowledge from the fermentation sector and applying it to the challenges that are very similar here. I want to open this up. I'll ask a couple of questions that are just sort of more broad, and any one of you can chime in on your thoughts. But we still continue to hear about animal serum and other animal components that could be used in media. Obviously, Paul sort of walked us through a nice history of at least how it's been eliminated in human iPS culture, but it's still a common ingredient for especially these new primary cell lines that need to be established. So curious about your comments, anyone, on sort of where the industry's at today in terms of using these entirely animal-free media components, and or some of the progress you've had in-house with eliminating some of them.
Sure, I'll jump in if you don't mind. So it was great to see that brief history of media development, because I was recently reading literature and came across one publication entitled something like "Why Do We Still Use Serum for Biopharmaceutical Manufacturing?" and was surprised to see it was published 22 years ago, because we're still struggling with this, right? And I'm not pointing that out to be pessimistic, but rather just to underscore that it has been a focus, and it's something that we still need to make progress on, given that many of the serum replacements or serum-free formulations now have been successful with pluripotent cells or immortalized cells, but there are some more challenging cells that this industry is aiming to work with. So I really see that there are three main bottlenecks in eliminating the serum. One is just the sheer effort it takes to convert over an established process, whether it's R&D or where you need to have continuity across experiments, or it's an improved, therapeutic manufacturing process. It takes that concerted effort and resources that can seem maybe duplicative, but so important. So I just think that there's a activation energy hump we need to get over, if you will. And then it's the proprietary nature of some of these serum alternatives that are on the market. And some of that, at least in our hands, are not always effective based on what cells you're trying to use, because serum is extremely complex and it has 1,000+ components, and the biology of cells, it can be quite different. So I think that's the third thing, is just the cell biology. I mean, really being able to understand that and which components from the serum are necessary for any given cell type. Because it's not practical to make a serum replacement that has 2,000 components. That would be great, and it would probably work very well, quite universally, but it's not efficient, and it's certainly not an efficient way to make media. So I realize that in my response I didn't offer any solutions, but I think that those are the bottlenecks, that we've made progress, but there's still some hurdles on the cell biology, and then just also the resource side, applying the dedicated time and effort to do it.
Anyone else want to chime in on that? Or
Yeah, I actually really want to echo what Aletta touched upon in the end there, the cell biology, understanding the cell's biology to be able to replace serum efficiently so that it does not compromise the yield or the cell density that you get when you have serum in there. So I think some progress has been made to reduce or completely eliminate serum, but that has come at a cost of cells not performing as well. So a culture is not giving you as high of a yield. So it really, I think the solution is to understand the cell's biology, and it depends species to species. But again, it's such a laborious, resource-intensive process that it's taking longer than it should or we want to. But yeah, I think serum replacement is more about understanding the mechanism behind replacing these with whatever nutrient cocktail we have.
Great, yeah, and I'd like to dovetail off the comments around the proprietary nature of some of the supplements and media formulations really in general. So obviously it's a really important piece of the intellectual property puzzle for companies. As mentioned, we need a lot of resources. There's a lot of novelty, especially with new species and cell lines. But that makes it so that there aren't that many formulations sort of off the shelf that are available for groups like David's that can, get into this sector in the academic realm and get going pretty easily. We also know that, regulators are going to ask for the components, to do proper safety assessments, and consumers may also be demanding to know exactly what's in there. So how do you, each of you reckon this sort of clash of incentives between, protecting medium compositions as a cultivated seafood manufacturer or a supplier, versus making them more transparent and available?
Well, I think, and I'm the academic here, but let me just say that, I think our goal is to get media that will work for general cell types and put it out in the public domain. And I really doubt that any of those media that we publish will be something that a commercial company will use, but it could be used as a starting point to get closer to start closer to the optimum. I think that's probably the key. Most companies won't try and patent their media because it's too easy to get around those patents. So, and it is what's going to make be one of the main things that makes their process good and commercial and make money. And so I would really doubt that those are going to become public knowledge in the future.
Aletta, is there any strategy you're taking with MilliporeSigma? Obviously you want to sell into the industry. So how do you figure out that target market?
Right. Yeah, no, it's certainly we have to strike the right balance and are open to, many different models. I do think it's important for everyone to be able to maintain some kind of trade secrets, for example, around the exact amounts of ingredients in a product or the nuances behind a manufacturing process that might play into some unique sensory aspects of the final food product. What I would hate to see, since David, you mentioned patents, I would hate to see patents that block cultured meat producers from using cell culture ingredients that have been used for mammalian cell culture for many years, right? Especially those that are being leveraged to eliminate serum in the processes. Although in very rare instances, our organizations have patented novel molecular structures of medium components. Primarily, we maintain trade secrets for exactly the reasons you mentioned, Elliot. But I will say that a significant amount of our cell culture media revenues comes from the manufacture of our customers' formulations. So that's exactly right, David, that it would be great to see, this base of knowledge and have publicly available starting points. But we fully anticipate that those will be tailored and optimized for any given process, because even a 5-10% increase in productivity by really slimming down the media and having just what you need for that process is significant. That's significant savings for the companies, so for the producers.
I think that's really key and good knowledge to know for others out there working on this challenge. I want to squeak in one more question, then we can go to the audience if there is time. But it's sort of, popular saying is you are what you eat, and in much the same way, right? The nutritional composition of these cells will be dependent on what they are eating or consuming during the process. So, how are each of you incorporating this line of thought into your R&D programs with, food being the end product in mind?
I think not only is the media going to affect the nutrition of the product, but it's going to affect the taste of the product. It's going to affect the stability of the meat product and so forth. So I think, and of course, optimizing for all those things is going to be nearly impossible. So I think the first optimization will be towards growth and differentiation. And then, further, after that starts getting to scale, there's going to have to be a lot more emphasis on, or at least the pilot scale, there's going to have to be a lot more emphasis on these other things, whether it's nutrition, sensory, or stability. And, in our group at Davis, we have people who will be working on all of those things as we get further along in scale.
I think at Wildtype, we see this as a two-part approach, where one is just to not add, or in a regular seafood, there are full of contaminants. And so the first thing is just avoid all of those contaminants. So we don't add microplastics in our culture medium or antibiotics. And so you avoid the things that have come to be in the nature that shouldn't be. And the second part to it is just have a nutritional equivalence. And for that, I think we just let nature guide us. And for us, it's important to not just have same amount of fat, it's important to have the same ratio of omega-3s and not have a skewed one there. So that's how we look at nutritional equivalence, or put your feed in the cells from what you would eat. That's our approach to it.
Great, thank you. And just a last quick question. I'll aim it at Aletta, since it was focused on media recycling developments. I know you had a call for proposals there. If there's any update you could say on developments related to media recycling or comments, and we can close it out after that.
Sure.
Yeah, you're back.
There we go.
Yeah.
Okay. Yep. I think I overclicked. Yeah, so we do have that call for grant applications. I believe that may have closed because we're now under the process of evaluating. So that's pretty exciting for us to be able to go through some of the ideas and provide some funding for this kind of research. I think that the challenge is going to be around, again, making it cost effective, and also capturing not only the larger molecular weight components, but also the smaller molecular weight components. I do think it's a big challenge, but stay tuned on the announcement of the winners there.
Excellent. Well, we are at time. I wish this session could go on for another hour and a half. I really learned a lot just in this brief time from the presenters. So I want to thank all the panelists and all the speakers for this session, for your insights throughout the panel. I remind participants to check out some of the resources that we have linked below, where you can learn more about cell culture media and cultivated meat. And thanks for attending.